My name is Madeline Brashears. I was a chemistry major at Bard. I worked with Professor Swapan Jain on my senior project, which was titled Synthesis and Characterization of DNA Functionalized Carbon Nanotubes and Their Interactions with Small Molecules. That's a lot to unpack, I'll be the first to admit it, but I'm actually positive that all of you have interacted with carbon materials in your life. The graphite in your pencil tip, completely carbon. The coal that we unfortunately burn for energy, carbon. Diamonds, all carbon. But this is a bit of misleading. I did in my title say carbon nanotubes, meaning that the carbon materials I was working with were on the scale of nanometers, or 10 to the negative 9 meters. That's really small. Carbon nanotubes can be conceived of as graphene rolled into a tube that results in a tube that is anywhere from 1 to 2 nanometers in diameter and several nanometers in length. You may have heard of graphene. It's a revolutionary material. It's 1 atom thick. It's fully conjugated carbon, which means it's carbon bonded in that honeycomb pattern you can see up there. That honeycomb pattern lends both graphene and carbon nanotubes interesting electrical and mechanical properties. And both of these materials can be functionalized for different purposes, such as energy storage and targeted drug delivery, to name just a few. What I did in my thesis was functionalize carbon nanotubes using DNA. Now, due to naturally occurring attractive forces between the bases of DNA and the honeycomb pattern of the carbon bonds in the carbon nanotubes, DNA naturally wraps around carbon nanotubes in a helical fashion, as you can see in this cartoon. The DNA is the red strand with the gray teeth. Now, what's really fascinating about these molecules, these hybrid structures, is that they fluoresce, which means upon incident excitation light, they emit a different wavelength of light, a different color of light. What's even more fascinating about these materials is that any disturbance in their structure will be registered with a change in their fluorescence. So if a small molecule were to disrupt the helix of the DNA wrapped around the carbon nanotube, shown by that cartoon yellow circle, the emission wavelength, the emission color of the fluorescence will change. If something were to degrade the DNA, similarly, the emission would change. So why do we care? Well, these molecules have interesting potential to be in vivo biosensors. So let's break that down. In vivo means in a living organism. They are biocompatible. Biosensors mean that they will react specifically to different molecules of our choice or design in the body and register that change through their fluorescence. So these molecules have been shown to be sensitive to hydrogen peroxide, nitric oxide, and even glucose and dopamine. What I wanted to explore in my thesis was whether or not they were sensitive to cisplatin, shown in that molecule there. Cisplatin is a common chemotherapy drug used to treat cancer. It targets DNA specifically and interrupts cellular processes to cause cell death. If I could show that these DNA carbon nanotubes were sensitive to cisplatin, down the line, far down the line, doctors could use them as in vivo biosensors to track maybe where cisplatin was in the body, how active was it, what concentration was necessary, and other questions like that. So that's like a brief overview of my project, and I can say that I was able to show that these DNA carbon nanotubes were sensitive to cisplatin after a 48-hour incubation, and that's a lot of data. And I have a lot of more data that you're welcome to ask me about. And you don't have to know what that table means necessarily. I would like to switch gears really briefly, though, to talk about just my experience of doing the senior project at Bard. Being able to explore this topic, I was actually the first to explore nanomaterials in the chemistry department, and the senior project and the independence thereof allowed me to do that. I also encountered many obstacles throughout that and developed amazing problem-solving skills, in my opinion. A lot of hard nights were overcome. And lastly, I gained a lot of valuable independent research experience. I don't think an experience like this would have been possible at any other institution, and I do want to acknowledge Bard for that and the independence I was given for this project. I do now know that I want to pursue materials chemistry and nanomaterials chemistry in my future career and PhD. However, I don't want to work with carbon nanotubes anymore, and you can ask me about that during the Q&A. Anyway, I'd like to thank you for your attention and pass it on to Charlie and Anaya. Thanks so much.